All right. Hello, everyone. Sorry I'm a few minutes later than I normally pop up here. I've been trying to get everything together, and students are very needy. You guys are fine. I have a lot of students who really need a lot of help to figure out what's going on with their lab exam and their grades, and do I have to drop? And so I've been working my hardest to make sure everyone feels comfortable. So we're gonna so today we will talk through the today at 3:30 we'll talk through the insulin activity. The next um, the next one I need to look into. I'm not sure which one we're doing at this point, but we're gonna figure it out. I know that we may end up doing a different kind of activity next week. I will get back to you on that, but we will be doing activity as part of class, and then you can do it later. You can turn it in later. Okay. So let me pull up what we have. Go. It's for later. Where'd you go? There you are. Okay. All right. You. Okay. All right. So what's going to happen for this part here where we're talking about RNA and DNA and translation, transcription, and all that kind of thing. Basically, I'm going to get as far as I can today and Thursday to talk about those sections. And then we'll talk through the other parts here. Okay. I'm actively unclear how to tell if there's anyone actually viewing me right now. Hello all, if you're viewing this. Come in late, it's fine. So let's make sure that this is set up right. Okay, good. All right. There we go. Next week is the test. Okay, that's what I was trying to explain here. The test is going to be through chapter 12, and if we can get through any of chapter 13 by Thursday, then great. If we cannot get through any part of chapter 13, then it's just going to be on to chapter 12. Okay, so it's going to be chapters 7 is the start, right? Chapter 7, that's the linkage and mapping. Okay, that's the part that we worked pretty hard on. Part of chapter 8, what the parts that I covered. And then 9, 10, 11, and 12. Okay, so I will be putting up a review for the exam. That's going to come out tomorrow. Okay. So the test. The test itself will become available on next Wednesday, and it will be available until Saturday. I'm shifting that time frame just so that you guys have a little bit more time to study, and we can have more of a review session next Tuesday and prep for the test. What it does mean is that depending on where you are in the class, you may want to try to finish your test early I won't be able to get to everyone's exam, obviously, and there may be some wobble in your scores, but I will be able to give you some better concept of how you're doing. I'm also going to, later this week, share a grade tool to help you be prepped for how you might do, okay? I'm just going to rearrange my screen here so I can see things better. Okay. Okay, the test will be Wednesday through Saturday. Next Friday, not this Friday, next Friday is, so next Friday is going to be the final drop date. And that's why I indicate what you need to do is if you are feeling concerned about your grade or you do the online test and you do badly, then I would suggest that you take the test earlier rather than later. Let me know that you need some feedback beforehand. I can go through and see preliminarily what your score might be and give you some feedback about whether or not you are on the right track. Again, the grade tool will help. I won't be able to give you exact, but if I see, oh, that's probably going to be near 50, then I can tell you. Okay. All right. We're going to finish up chromosome structure and DNA and start on our transcription translation part. We'll start on transcription and RNA and the code. I'm going to introduce the code, chapter 12. Chapter 12 goes really heavily in detail, but I think that really understanding the code and how it's read is a better topic for the translation part. So we're going to jump around a little bit between 12 and 13. Really, we're going to start out by talking in chapter 12. The main thing that we're going to be starting on is talking about the coding dictionary, 12.4, through the transcription in transcription and coding. Okay, so I'll walk through which sections we're going through. All right, let's start this so I can start getting through it. Again, what we'll try to do again is to get through everything that you're going to be tested on by Thursday. On Tuesday, we'll have a review session, and then on Wednesday, your test will pop up. Okay? All right, let's start on this. So this is something that we are continuing. We talked about the types of chromatin before, euchromatin, heterochromatin, and that heterochromatin is either constitutive or facultative. That some of the heterochromatin areas can bind or unbind. That euchromatin is always able to be read, and heterochromatin is sometimes able to be read. So binding these genes can influence their availability for actual transcription. Okay, we're going to talk about this more as well with the RNA transcription because there are things that influence how RNA moves around. Okay, so again, euchromatin versus heterochromatin. We've had the less condensed. It's on chromosome R's. Lots of unique sequences. Lots of genes, often transcription. Note the heterochromatin is not never, but infrequent, and it does require some modification. Chromatin is more condensed in the heterochromatin areas. Again, near the tips, those are the telomeres, and near the centromere, where there's no crossover. All right. So I said I would expand a little bit on the polytene chromosomes I have. So these are giant double homolog chromosomes. So they have these active genetic sites that expand to give these puffs of chromatin. So these active sites are basically huge areas that are puffed out to encourage extra transcription. There's thousands and thousands of DNA strands in them. DNA of paired homologs undergoes many rounds of replication, and it doesn't have any strand separation or cytoplasmic division. It stays all in one cell. So you ask yourself, okay, when then would we be looking at this? Why would you ever have this? What would be the purpose of having a polytene chromosome? these giant, huge piles of chromosome of data, that there's thousands and thousands of copies of the same gene over and over in all these DNA strands. What might be the benefit of having that? Now, this isn't in any vertebrates, I'll tell you that. Only in a few eukaryotes. Anyone have any ideas? Why have this polytene chromosome? Why have thousands and thousands of the same DNA? Yes, to allow for some redundancy. What purpose could redundancy have? Why might you want redundancy? There's a piece of that is redundancy. Let's move forward, okay? They're common in Drosophila, okay? So the puff regions have localized uncoiling during genetic activity. Basically, when it's building stuff, it uncoils these areas and it makes them available for transcription. So these puff regions are the visible manifestation of very high genetic expressivity. So we talked a little bit about expression of genes. A specific expression of a gene would be that you can visibly see that there is expressivity of the specific set of genes. Okay, that's what the puffs are. They literally are puffed out, open areas that RNA can access at any time. This is the key. Drosophila uses this in the larval stage. Salivary glands create the pupal shell, and they have to cover their entire body with their hard pupal shell. And so at a specific stage in their life, they create these polytene chromosomes. And the puff regions that have thousands and thousands of these open areas encourage heavy production of salivary proteins to create their shell. So this is an interesting pattern here. Again, this kind of shows how the structure of chromosomes can influence the actual reading and production of proteins and genes. So that's an interesting piece with these polytene chromosomes, right? So these are the larval salivary gland cells. Again, you'll see they're really long and they're stuck together, and you see how thick they are. Again, thousands of strands all together. Each band is going to represent a gene that can be read. And so these are all salivary genes working together to form the proteins that will make the pupal shell. And this is what the puff looks like. Again, it's unwound at the areas that it wants to be transcribed. So there's high access to genes by RNA transcriptase. We're going to talk about RNA transcriptase and its importance and how that's regulated as well. Okay. So the next thing we're going to talk a little bit about in this section is chromatin remodeling. So this is what we talked a little bit about epigenetics, the idea that your expression of genes changes throughout your life due to the way that your DNA interacts with the environment. So DNA can be inaccessible due to interactions with the chromatin fiber, these histones, the winding type versus not winding. So in order to get DNA to protein, to get that from DNA, you create protein. Chromatin structure has to change. The idea is that while DNA may never change its sequence in your body, chromatin can change to allow access to different genes or not to different genes, depending. So basically, some genes must be able to turn on or turn off. So you have to have a way to relax the compact structure of chromatin, and you have to have a reversal mechanism to inactivate genes, especially if these are genes you don't use very often. Obviously, you wouldn't like it if, for a male or a female. You wouldn't like it if the produced milk was turned on, right? That would be pretty crazy. You have to have it turned off in certain regions of life. Okay. So chemical modifications to the actual chromosome structure is important for genetic function and for actually having certain gene segments available for transcription and translation. Okay. So histone tails, the tails of the histones that wrap around that the DNA is wrapped around. It's a potential target for chemical modifications. These histone tails, or the histone tails being closed, that's when the heterochromatin, open is the euchromatin. But you can change that through acetylation, methylation, and phosphorylation. Okay. All three of these modifications are reversible, meaning that you can turn on and turn off genes. Again, this leads to some thoughts about, well, how could we actually modify and edit our actual genes themselves by influencing chromosome and RNA reading of certain genes. So acetylation, it adds an acetyl group to the positively charged amino group on the protein. Okay, so it changes the net charge of the histone protein, of the chromatin protein. It neutralizes the positive charge. Okay, so if you acetylize these, you can remodel the chromatin fiber overall. Basically, you're rebuilding it. You can either increase the regions of active genes or decrease the regions of inactive genes. This is the weird bit, is that acetylation, often it is turn on a gene, but it's not always turn on because the structure of chromatin is so complex. Methylation. It adds methyl groups to the arginine and lysine residues and histones, okay? This can either, again, here's the weird bit, increase or decrease transcription. Depending on where they're added, they can either stop RNA from turning into RNA, or they can increase RNA transcription in a certain area. So you can increase the amount of RNAs that's being created and therefore the amount of proteins, or you can decrease it. But methylation, depending on where it's added, can do either, which is pretty weird. That it really does matter where you put it. Okay, so phosphorylation, that it adds phosphate groups, and this the phosphorylation actually induces the condensing of a chromosome. So changing that phosphorylation can change basically the way that chromosomes condense, but it's not as much into the gene reading part. Okay, so so this gives you a little bit of the chromatin structure and gene activity and some of the influences here. Okay, so DNA itself is wrapped around these histones, so it has its own negatively supercoiled. You add these core histones, and then these core histones are on a string of chromatin. And if you add more fibers together and wrap them around really tight, and you close these tails on the histone, then you get less active genes. Add further scaffolding proteins, which the phosphorylation does, that condenses the chromosomes. Again, you have to be able to uncondense them. The ones that are in the condensed form are not being read in any ways, so you have to be able to uncondense it, and that is the reversal of the phosphorylation. So it's an interesting pattern here. You have all of these ways to change things. Okay. So now I talked about that a little bit. What would happen if we neutralize the positive charges on the histone proteins, which would be the acetylation? What normally happens to the DNA? Anyone there? So if I went too fast, we can walk back. So neutralizing the positive charges, again, this is acetylation. Mavis says A. Anyone else have an answer? Think it's a different answer? Faye says A. All right. I guess no one else is going to answer right now. Neutralizes the positive charges. Yes, Mavis. Okay. The key here is the separation. So let's go back real quick. Again, the histone tails. I mentioned that adding the acetylation can change the net charge of the protein. It remodels the chromatin fiber. So acetylation, what it does is actually neutralizing the positive charge means it no longer sticks to the DNA. DNA is negatively charged. DNA is an acid. When they are bunched up and coiled, okay, let me go back. All right, here we go. Acetylation pulls them away. It neutralizes that positive charge. It lets them go. Okay, DNA becomes accessible when you acetylize that, but it can decrease the activity of already inactive genes as well, because those inactive genes are influenced by what the shape is around here. Okay, so if there's a mildly active gene near the centromere, then releasing these can actually reduce its activity more instead of make it better. So it's sort of a complicated piece. It's hard to tell. It depends on where you're working in the chromosome, okay? All right, let's move back forward here. All right. Again, separating the DNA, that's acetylation, okay? If you neutralize a positive charge, it no longer sticks to the DNA, and that means it separates, and that means the DNA is more accessible. But depending on where you do it, it can actually, it either increases activity or decreases activity, but it has to be active versus inactive genes. All right. So heterochromatin. Who remembers how heterochromatin works? I'll walk through this real quick. Anybody? Answers? Heterochromatin is characterized by all the following except it is present all over the inactive X chromosomes of female mammals. It contains genes that are transcribed at a high level. It's made of highly repetitive sequences. It permanently condensed through the cells, present telomeres, centromeres and telomeres. Okay, I see a couple of B's. Anyone else have any other ideas? I think D's. All right. The deal is that heterochromatin actually does not contain genes that are transcribed at a high level, because it is true that it's present in the inactive X chromosomes. It's made of highly repetitive sequences, and the idea is that with inactive X chromosomes, it's almost permanently condensed. The idea is that it contains genes that can be turned on or have a very low transcription level. Okay? It's not like there's some central force that's saying RNA only talk to this DNA molecule. If there's DNA available to talk to, RNA will talk to it. So it's making the DNA inaccessible to stop the transcribing. Again, this is why triple X people, XXY, X0, those people have different expressions, because the inactive X chromosomes with heterochromatin, they contain genes that are actually transcribed at low levels, but they're not always completely turned off. And depending, sometimes your body can turn it back on. All right. So that's all for chapter 11, okay? I just wanted to talk mainly about the chromosome structure, how that influences how the DNA is structured, and how RNA can access it. Okay, so those are the main things I wanted you to get from there. I'll make sure this is up. I thought I put it up, but I don't remember if I did. I'll put it up. I'll make sure it's up. Okay. So this one is new, and I'm still working on all the pieces of this one. Okay, so it's a work in progress. It's still being made, but I want it to mainly. Oh, that's not what I want to do. Exit. Okay, no. I want to make my slideshow work better. Here we go. Do this so you can see my pointer at all times. Okay. All right, so we're working on chapter 12 here, okay? I know that a lot of you have gone through transcription. One of the main things I'm focusing on here in, okay, good. Beatrice says I did put it, I couldn't remember if I put that second one up or not. There's a lot of stuff I'm trying to keep track of with many classes, okay? So transcription overall, okay? One of the main things I want to be focusing on here is how we're going to be focusing a lot on kind of the concepts about how RNA and its interactions with DNA influence what genes are actually read, at what level they're read, and how intense their interaction is. Okay. And so some of this you've already gone through. Some of this we understand. Okay. The transcription and translation periods here, so that's chapter 12 and 13, those really have to do with how RNA creates proteins. And really understanding how RNA creates proteins helps us to understand how gene expression works. So we're moving away from just talking about what do the genes say, and now we're working on how does the cell interpret these genes and create the ones that it needs to in the proper way in different cells, in different tissues, etc. Okay? So now we're talking about the actual movement of gene to protein. This has to do with expressivity. So we talked a little bit about you can have the genes and not express the traits, and that's because there maybe is a low penetrance or low expressivity. So the expressivity part really has to do with RNA and DNA interactions. All right, this is the central dogma, obviously. We have talked a little bit about DNA replication. Now we have transcription, DNA turning into RNA, and translation, RNA turning to protein. All right, so DNA is the template for the synthesis of RNA, so we're going to talk through that and a little bit about how RNA chooses in a way to interact with the different things. So how the chemical pieces work together. Again, this is a little bit of biochemistry, but it's a little bit of gene expression. All right, so unlike DNA, RNA synthesis is unidirectional. A single strand serves as the template. We're going to talk a little bit about the creation of the various types of RNA as well. So DNA replication, you transcribe the entire strand, but only certain parts of the chromosome are transcribed when RNA is done. Remember that certain parts are have that methylation tightly wound in the heterochromatin and such. Right, the chapter 12 PowerPoint. I'm going to tell you that I'm working on it actively right now. This is the one you're looking at, and I am picking it up and putting it in there after class. So this is all new. I have just built a lot of this today, so I just want to make sure that this was put together. So a lot of this is coming up. You're going to see it. You will. Okay. All right. One thing that your chapter 12 actually starts with is talking about the genetic code. I'm going to introduce the genetic code so that you just understand what mRNA is trying to pick up. And you should know some about the genetic code. I mean, we talked about this a little bit. But for the genetic code overall, we want to discuss a little bit about how the genetic code interacts with RNA, and so how RNA is reading it, and then what each of these does. We're going to go over it in more detail, exactly how these different sections are recognized by the ribosome when we talk about translation. All right, so generally, the genetic code overall is written in linear form using ribonucleotide bases that compose mRNA. Okay, so this is RNA, ribonucleic acid. This is a linear thing. There's also nonlinear RNA, and we'll talk about that a little bit. But each word, which would correspond to a specific DNA, specific amino acid, that consists of 3 letters, the triplet code. Okay. Codon is every 3 ribonucleotides. That together is the codon. Okay, the individual word. The key here is that the genetic code is unambiguous. Each triplet specifies only 1 amino acid. And it's also degenerate. Degenerate means that a given amino acid can be specified by more than 1 triplet codon. Okay, also known as redundant. Okay, so it has 3 or 4, and I'm going to walk through why it has 3 or 4 in a minute. So it contains start and stop signals. So these triplets that are ones that initiate and terminate translation. These are important because it tells the RNA basically, for one thing, it tells the other proteins where to start. Where does the gene even start? So certain things on the DNA actually tells the transcription process to begin. And then you have the start and stop signals near the end that tell the RNA to stop transcribing, and for RNA to terminate translation. Okay, so they're commaless. Basically, once translations begin, codons are read with no break. We talked a little bit about some of the different types of mutations, and one of the biggest mutation issue is that when a single nucleotide is inserted or taken away. Because inserting one single nucleotide can shift the entire reading frame. So it's not like it's checking and rechecking that this is the right thing to read. It just reads from beginning to end. So if you take out one of those in the beginning, then you can shift the entire code and make a whole string of things that are wrong. All right. So it's non-overlapping. Any single ribonucleotide within mRNA is part of one triplet. Okay. It's also nearly universal. A single coding dictionary is used by viruses, prokaryotes, archaea, eukaryotes, anything that has information in it. Uses this coding dictionary to create. The COVID virus, as we talked about last week, the COVID virus actually uses the process of RNA duplication, RNA transcriptase, in order to read its own DNA and build it inside of our cells. Okay, but it still uses the coding dictionary. So that's why, viruses can infect us and still make their own proteins because it's the same coding dictionary. All right, so we'll talk a little about the genetic code. Again, we're going to cover a little bit more about recognition and RNA movement and such when we talk about translation, but let's just work through the types of codons here. So there's sense codons, encoding amino acids, initiation codons, and termination codons. Okay, so all the rest of these, every single one of these in green, is a sense codon. UUU and UUC, that's phenylalanine. UUA, UUG, that's leucine, etc. Okay. There's a couple of them that say stop. That means stop creating a protein. These are not stop reading the RNA. We'll talk about what tells the RNA to stop and what tells the RNA to start. But you should have learned a little bit about these already. But then the AUG, that's always the beginning of it. That's the one that tells the ribosome to attach. Okay. And there are 64 possible codons, but there are only 20 amino acids. So these are things that you probably already learned. But the idea is that multiples code for there are multiple codes for one amino acid. Okay, so the genetic code is what's called degenerate. Each codon is written as it appears in mRNA, reads 5 to 3. Okay, so we're assuming that this is the 5 side, this is the 3 side, and that's how it's read down that edge. So a degenerate means you have that anything that starts with UC is going to be serine. You have the UCU, UCC, UCA, UCG, etc. So this has to do with something called the wobble hypothesis. Okay, in that when it's being translated, it can wobble, and so the first two are the most important, the first two, and then the last one can wobble back and forth and still produce the same RNA. Okay, this means actually that the tRNAs can they can match up to 2 of the first, and then it doesn't matter which one it has. It's going to have the same. It's going to have the right amino acid. Okay. So these are synonymous codons. They specify that for the same amino acid. All right. So while each ribonucleotide in RNA is part of only one codon, there can be overlapping genes, even though the genetic code is not overlapping. Okay, so a single mRNA can have multiple initiation points. AUG can happen at multiple places, creating different reading frames and specifying more than one polypeptide. That is a bit rare, but it can happen in that you can have one mRNA with multiple initiation points, reading it a couple different ways, which makes it for an interesting thing. One genetic code could tell you to do more than one protein. And that's part of that wobbling as well. All right, so this is the basics here. We have DNA. This DNA base sequence triplet of a gene codes for synthesis of a particular polypeptide chain. Okay, so they have these triplets. And then you code for the base sequence of the mRNA and for the consecutive base sequences of tRNA as well. Okay, so tRNA tells us, so it matches here, and then this attaches to the ribosome. Okay, we'll walk through the attachment so this matching and attachment when we get into translation. But I wanted to focus on this piece in particular. We've already learned a little bit about transcription and translation. I know you guys have some base knowledge of this, but I think one of the key things is understanding how that impacts gene expression too. How does translation impact what parts are read, and how does transcription change what proteins can be manipulated with a single mRNA? All right. So I'm gonna, again, there's a lot more at the beginning there that talks about the genetic code and how they find the code and how they figure it out. Those parts I'm a little bit less concerned about. At this point, what I want to get out of this section is understanding really how RNA interacts with DNA and how it tells what it's supposed to do. Okay? So ribosomal RNA is transcribed from genes. mRNA is the only one that is constantly going in and out. That it goes in and it goes over to the RNA. But this RNA, the ribosomal RNA, it's built via mRNA and transcribed from genes just the same way. And then that's built into the protein part here. Okay, so it has a small subunit and a large subunit. So this translation requires the assembly of large and small ribosomal units. So we have RNA, rRNA, and proteins. Again, this is built via the same gene process. We have the DNA to mRNA to the ribosome. At some point, the first ribosome has to be built, right? But a lot of those ribosomes come in over from your mother's side, and then it's built from the continually built from the information inside the genes. All right. So again, the first mRNA, the first ribosomes are in your mother's egg, and then once the new nucleus is formed, then information mRNA from that comes out, is read by your mother's ribosomes, and can then build new ribosomes. Okay. We have transfer RNAs, tRNAs. So these are transcribed from genes as well. So it's a linker molecule between mRNA and the protein being produced. Okay, so again, these kind of tRNAs, it's the same concept here. Once it comes off of once it comes off of the genetic code, it comes out, and when tRNA is created, it immediately begins building. It forms into this shape and then comes out and searches for the amino acid in the cell. Okay, so there is actually no protein as part of these transfer RNAs, so it doesn't have to be built like a ribosome does. Instead, you have this transfer RNA. It's transcribed directly from the gene, and it forms in that shape and picks up amino acids and goes out to the ribosomes after that. So it's a linker molecule between mRNA and the protein being produced. Oh. Something happened there. Okay, so it reads the genetic code off the mRNA. It brings the correct amino acid to the ribosome. Has a distinct low-relief shape. One of the key things here is that it's highly stable. RNA very stable. Okay. Attaching RNA to proteins and putting RNAs in these cloverleaf shapes. Okay. So you'll see these CCC, GGG. Again, this is read from a specific section in the DNA. Once it's read, it forms into this shape, and it has this little amino acid attachment site. It's always a CCA site, but different parts of this will code to attach it to a different base. Messenger RNA is what we're going to be focusing on a lot. Okay? It's transcribed from genes, but it's not as stable as the other two. It carries biological information stored in the DNA to the ribosome for translation. It is not attached to any proteins, and it doesn't have a secondary structure. It's a linear molecule, which makes it in trouble for degradation, just like our linear chromosomes have issues with the ends. It's unstable. It's transcribed, it's translated, it's degraded, and this is because it has to be edited and built and read and moved and interacted with. This is the most unstable form of RNA because it is transient. The goal is that it needs to just survive long enough to be read by the ribosome, and then it breaks up and a new mRNA would be formed in the nucleus. All right. So we're going to talk about transcription in particular at this point. After we talk about transcription, we're going to talk about the editing of mRNA and also the creation of tRNA. Okay? So the process here is we're going to go through, so we already talked a little bit about the genetic code, the coding dictionary, different initiation points. We're going to talk now about how RNA transcription synthesizes RNA and RNA polymerase directs RNA synthesis. So that's 12.8 and 12.9. And we're going to talk a little bit about how bacterial versus eukaryotic transcription varies. That's not as important to me because again, we're mostly talking about gene expressivity. We are going to talk a little bit about the processing of RNA in particular. RNA is processed in eukaryotic cells and not processed in prokaryotic cells. There's no reason to process it. This is read right away by ribosomes, but it is processed in eukaryotes. But we're going to talk about introns and exons too. Okay. All right. Transcription synthesizes RNA on this DNA template. The production of mRNA is what we're focusing on right now. This is the intermediate molecule, obviously. Okay. So RNA polymerase is an enzyme that directs synthesis of RNA using DNA template. Remember, RNA polymerase is the same thing that's functional in DNA replication. It's part of that primase that comes over and puts that starting part to the eukaryotic. So it puts that starting primer to start the DNA. The DNA replication, okay? But RNA polymerase itself doesn't need any primers. It's very reactive. That's one of the key things about RNA is that it can be reactive. So it has the same substrate requirements as DNA polymerase. It has to go from 5 to 3, but in this case, it doesn't have an ends issue. No primers required for the initiation, and it doesn't have to remove sections, or it doesn't have any issues with the leading and lagging. It just goes from 5 to 3. That's it. Goes straight. Transcription is started. So that's called the transcription start site. It begins, and the DNA double helix is denatured via RNA polymerase. It has a reaction to itself. Oh, there's an issue with my connection. Is anyone having trouble hearing me? I can slow down and go back if there's an issue with the connection. How are we doing? Okay. Perhaps there may be an issue on Angie's end. Again, I'll slow down if I need to. But Angie, this will be recorded. I'm recording it via my computer, which doesn't have to have internet to do that. So it'll come through pretty clearly in the recording at least, even if it doesn't come through clearly for you now because of any issues. Okay, good. I'm glad that we are not all having issues. I've been looking at my program, so looks clear here. Okay, good. Gotta double check that. Okay. All right. Go back. There we go. Okay. So RNA polymerase interacts with a couple of other proteins in order to tell it where to start. So one of the interesting bits about gene expressivity is how promoters influence the efficiency and the intensity of transcription in a certain area. Okay. So promoter sequences, they govern, again, the efficiency here. So RNA does the same thing, okay? It goes 5 to 3, so starts at this 5 end. It builds out to, it builds out this way. It's building on the 3 end here. Okay, so this 3 free end is being built on. Build. And then it comes off. So it doesn't, again, it's not transcribing the entire molecule. It has to be told where to do it. Okay, so you have a template strand that's being read. One of the strands is always the template strand. One of the strands is always the non-template strand. And we can tell which one is a template just due to a certain DNA sequences that tell it to attach in a certain spot. Okay. Initiation of transcription. How does the RNA know where to attach and how does it know what it needs to do? There's this promoter, something called a TATA box. Enhancers and there's silencers. Okay, so the TATA box is this repeating section of TA. I think it's actually a TA, something similar to that. Okay, so the idea is that there's a protein that it binds this at a specific section of the DNA. That's how we can also tell the location of some of these starts is where the TATA box is. Okay. So as a promoter section, again, this is the TATA box. This is where RNA transcription begins. RNA polymerase will attach right here. It's going to read it. And then there's a segment here that terminates. Termination varies between prokaryotes and eukaryotes. The key here is that usually then there is after the end here, again, it's not necessarily, the RNA isn't told to stop, it's told to stop at external parts in eukaryotes. There's a terminator sequence. this sequence is not going to be read at all by the ribosome, because right here is where you have a stop codon. And then after that, it's terminated. And an external part comes over here, so we'll talk about some of the pieces that tell it to start and stop. Okay. One of the interesting pieces, though, is that the promoters and there are control elements that talk about promoters. Again, you have these initiator proteins, okay, and they attach to the promoter segment, all right? Before that, on the DNA, though, there's these control elements. The control elements attach different pieces, and depending on what's attached there, it will tell the RNA to continue to keep coming, or to tell it just to transcribe it at a normal level, or transcribe it at a high level, or to repress it. So I want you guys to look at this, and one of the things we're going to be getting into after the second exam, that before we end, is talking about some of these new gene therapy pieces. How could some of this information about what RNA is told to do via outside proteins influence how we might do gene therapy? I want you guys to think a little bit about that. Could something that has to do with RNA transcription influence how we would approach gene therapy for people who either want to turn off or turn on a certain gene? Talk a little bit about the DNA structure, but what about RNA and transcription? Anyone have any ideas? So again, what we're trying to understand here is looking at this information, okay? These are these promoter factors. This is the start point. Past here is the gene. This says to RNA RNA polymerase, start here, start reading, okay? Start reading in this direction on this strand. So it attaches to that. It opens up the gene here. But how could this information tell us a little bit about, again, gene therapy, changing the way that our bodies interpret the genes that we have, the way that our bodies use the genes that we have? Any ideas? Anyone following what I'm trying to get at here? Confused what I'm asking? All right. Okay, Mavis is confused. All right. So, in DNA, there you go. Okay, Epere has some concept here. The concept here is that if you have, let's say, an error in your DNA, and you're producing a protein that is harmful to your body, that potentially, if you could have a silencer protein that is targeted to that specific gene that is causing the bad protein to be produced, if you could put a silencer on that, you could change how you express the genes that you have. So let's consider what would happen if we were able to. Now this is not something we can do exactly right now, okay? But remember, the people who are heterozygous for sickle cell, they have somewhat sickled cells. They have somewhat normal cells. Okay, so that is not fully dominant. Okay, maybe co-dominant in a sense. What if we could turn off the sickle part? That would get rid of some of those issues with having partly sickled cells. Now, what if the issue is that someone with an incompletely dominant gene needs to just produce more of a protein. What if you were able to take a protein, target it to a specific gene, and put it in that activator site? Put an activator protein onto there, and suddenly, instead of having, you get rid of the issues of having only one copy of a gene you need. You're producing more of a good gene, a good protein, maybe less of a bad protein. Changing the way that RNA reads our genes is one way that we can actually influence our genes outside of changing our own DNA. Now, we'll be talking a little bit about DNA and RNA and the interactions of lots of different pieces of recombinants. But outside of that, just expressivity of genes can be influenced if we change the way that, if we are able to highlight activators and repressors and encourage them in certain people with issues, either low production of a good protein that they need, high production of a bad protein that they don't. There have been some studies that have shown that certain medications can induce, say, the reduction of a protein that is linked to, say, Alzheimer's disease. So some of these can be done through medication and not just, so repressing through a specific influence of chemicals in your body versus repressing via targeted repressor proteins. I thought that was interesting to point out here. This is one of the key things I wanted you to get from exactly how this is regulated. All right. So when RNA is transcribed from a gene, which strand of DNA is most often used? Specific strands are named certain things. Okay, so we have a couple of A's. And overall, that's true. The idea is that there's a template and non-template strand. The template strand is called that because it's the one that's used as a template. Either strand can get you a full DNA molecule, but only one of the strands is read 5 to 3 to identify that. And again, there are markers on the DNA that indicate start here, the initiation factor, start here, and start on this strand in particular. It doesn't attach to the other strand. It pulls it apart. RNA polymerase comes through, and then there's some information about where to stop. Okay. So the transcription unit overall includes all of these, except for an enhancer. The idea is that the transcription unit, it connects to the promoter, and there's a coding sequence and there's a terminator sequence. The enhancer is outside of that, and that enhancer, again, can be manipulated through environment or medication, or it can be manipulated to produce more of or produce less of. What's interesting is that these enhancers, again, our bodies do them all the time. You have certain, there are certain things that encourage us to produce more dopamine. And if you have trouble producing dopamine, well, there's medications for that that can encourage the enhancers to attach to your dopamine genes and produce more dopamine. So it's an interesting pattern here, okay? So if you have dopamine issues, depression is one of those dopamine errors in your brain where you're not producing enough, even if you have the genes for it. We have things that can turn on, that can enhance, and that encourages the enhancing proteins, okay? But all of those are outside of the actual unit of transcription. That's part of the sequence of DNA to encourage the reading, transcribing. Okay. All right. So if the bottom strand is the template strand, let's figure this out. Okay. I want you guys to understand a little bit about how it's transcribed, understanding what piece. Okay. So in which direction is the RNA going to be synthesized? remember you have to think about the fact that RNA has to attach. It's going to put down the first ribonucleotide, and it has to go towards the 3' end. So at the bottom strand is the template strand. What direction is the RNA going to be synthesized? All right, so one of the interesting pieces here is it's a little confusing. You're saying left to right. Now I want you to imagine that I attach the RNA on top of this template strand. If I attach it on top, then, and it starts to go right, wait a second, on top, remember it's going to be anti-parallel now. The 3' end is going to be on this end like this one is. It's going to look like the template, like the non-template strand when it attaches. It can't go right to left because it can't attach things to the 5' end. It can't go 3 to 5. It's going to have to go, sorry, left to right. It has to go right to left. Okay, right to left. That's the key. Okay? Start right, go left, because once it puts the first one here, okay, it's going to look like the non-template strand, except it will not have, again, the RNA transcript is going to look like the non-template strand, except for, obviously, these T's are not going to be there, okay? You have U's instead. But here's the idea. It's going to go U, U, and then A. Okay? UUA is going to be the first one that it does. It's transcribing this way because, again, once it gets up here, this is going to be the 5' end when it puts that first T on. And then there's going to be a 3' end this way, and it attaches new nucleotides to the 3' end. Okay? The template strand is the bottom strand. The non-template is the top. But remember, this is going to open. Okay? It's going to pull down. It's going to open up. And this is where it attaches. So it's going to look like the non-template strand. And it's going to go in the direction 5 to 3 that the non-template strand would reproduce in. Okay? Key here again. This is 5 to 3, but when it attaches, it has to go 5 to 3 on its own molecule, own direction here, okay? When it attaches, its 5' end will be on the right. So it has to attach, grow on the 3' end and go 5 to 3. So it's going to go right to left. So just understand that it's doing the same thing that DNA would do as well. So this is the same answer for a DNA problem. Okay. So transcription requires that the DNA and RNA knows where to pair. Okay, so that's part of that promoter and the enhancer and all that. Okay, it's actually 5 to 3. The transcription is always adding to the 3' end, so it's going away from the 5' end. So it's 5 to 3. It's not 3 to 5. The 5' end is unreactive. The 3' end is reactive. You must add new nucleotides to the 3' end only. That way it goes from 5 to 3. 3 being the reactive. Okay. Do you understand what's happening here with Mavis? Okay. If it's going this way, this is what's showing. It's showing us building this way. This has to be the 3' end. Okay? Starts at the 5' end. See, it says 5' here. 5' will dangle out because it's already been done. 3' end is where the new nucleotides are being added. That's always true, including for DNA replication. Always added that 3' end. But the DNA itself, this DNA, this is the DNA's 3' end over here. This is the DNA's 5' end. Okay? It flips because it's on the opposite side. Because it's going to be anti-parallel to the DNA itself. 3' end of DNA, of the RNA is here. The 3' end of the DNA is on the other side. Okay. This is RNA polymerase. This is just a 3D model of the actual protein. And it's attached, it kind of hugs and holds onto the DNA and spits out the mRNA this way. Okay. So bacterial RNA polymerase, we're going to talk about both bacterial and we're going to talk about bacterial RNA polymerase, and we're going to talk about eukaryotic RNA polymerase, okay? So what they have here is this core RNA polymerase, and then what's called a hollow enzyme, okay? So this sigma factor will bind to the promoter and initialize the transcription. That's what I was pointing out. This is basically the initiator protein, that it binds to that promoter. The promoter gets this factor. This happens both in DNA and RNA, that there is a promoter sequence on the DNA that a protein attaches to, okay? So it's called a sigma factor in bacterial RNA polymerase, okay? So this is the only two states it does, okay? Either the core RNA polymerase, and you have the sigma factor that attaches to tell RNA polymerase where to go, okay? So that initializes the transcription. All right. Again, you have this promoter, and this promoter piece here, so it tells you where to start. That sigma factor will leave as soon as the RNA comes in. It replaces the sigma factor, okay? Sigma factor helps open it, and then the RNA polymerase grabs onto it and starts reading the template strand, okay? Again, it's going to be reading it on the 3 to 5 oriented strand, so it can go 5 to 3, okay? Starts reading it and growing. The 5 end sticks out. The 3 end is being built. And then there's a terminator sequence that tells it when to let go. That's the basic pattern, okay? So the sigma factor would then bind to the RNA. It will go find a new spot to attach, attaching the RNA with it, and continuing, okay? In eukaryotes, different types of promoters are recognized by different types of RNA polymerases. Remember, there are different types of RNAs, and they're all transcribed from the genes, okay? So RNA polymerase 1, 2, and 3, depending on what the gene is signaling to make, will attach to different promoters, okay? The main promoter in eukaryotes and prokaryotes is that TATA box. Mostly that's making mRNAs, okay? So when it says pre-mRNA, so that pre-mRNA is before processing. We'll talk about mRNA processing on Thursday, hopefully. But the idea is that RNA polymerase 1, so that has a promoter that only recognizes ones that will transcribe rRNA. RNase polymerase 2 is the major one that's being used to create mRNA. And then there's these other microRNAs. Don't worry about these SN, SNO, okay? So those are just these mini RNAs that sometimes work as, they target certain proteins to be transcribed. This is the main one. Pre-mRNA, the non-processed mRNA, is what RNA polymerase 2 is mainly doing. Then you have RNA polymerase 3. tRNAs, and any of the smaller rRNAs that would make up the ribosome, okay? And then a couple of these other extra ones. We're mostly focusing on these, okay? But these 3 are the ones working in eukaryote RNA polymerase, okay? Each of these is creating a different type. Mainly RNA polymerase 1 for large rRNAs, the ribosomal RNAs. A different promoter will say, hey, this is a sequence to make RNA, and RNA polymerase 1 will attach to only those sequences. RNA polymerase 2 mainly is for the messenger RNA. RNA polymerase 3 mainly for the tRNAs and some smaller rRNAs. Okay, so those are one of the key things here in the eukaryotes. It divides those apart, while in bacteria and other prokaryotes, it has only the just single type of RNA polymerase that it recognizes all types. Again, it has a much smaller genome than we do. And while they create rRNAs and mRNAs and tRNAs from those, there's no differentiation. It's just a single RNA polymerase versus we have 3 to each find a different spot to be reading. Okay. All right. So I'm going to skip this part actually. I want to focus on this part. Here we go. Okay. This part is from a different book actually, and I don't think we're not going to cover this kind of consensus sequence sort of concept here. Okay. Let's talk about the stages of transcription. We've already encountered this a little bit. I know you guys have seen this before. Again, we have that initiation, the DNA promoter sequence. You have a protein attached to the RNA polymerase that attaches to the promoter and helps open up the DNA, and then RNA will transcribe. The RNA polymerase will then unwind the DNA with the help of the initiator protein. The new RNA is synthesized 1 nucleotide at a time, 5 to 3, and then you have the termination. Okay. So this is one of the parts where I show you exactly where the promoters are. One of the keys here is that as we have been able to read the entire human genome, some of the information about where the genes start and end is based on these TATA regions, okay? So prokaryotes have this TDGACA, okay? Minus 10 means it's minus 10 down, okay? This is where the promoter region. Further upstream of that is where you'd get those extra silencer or enhancer proteins that would attach. But if we see this segment, this is not in a lot of actual genes. This is telling us, hey, a gene exists here. This is where RNA is transcribing. We can also, through the some molecular biology, basically see where the proteins of RNA polymerase are attaching. But some of just getting the information about reading the entirety of the human genome, we can see the beginnings of certain genes by looking for this TATA region, okay? It says TATA. TATA. All right, the promoter indicates where transcription should begin, which strand is to be read, and in which direction the RNA polymerase will move. Okay, so it's telling you which one to, so the TATA is on the non-template strand, so it attaches to the other side, but the idea is that the RNA polymerase detects the TATA region on that side and opens up and attaches on the other side. Okay. And because now where it attaches, it determines which way it's going to move. Based on the direction of this TATA, okay, again, it doesn't read the same way either way. It tells you it's going to move this way versus move the other way, okay? It can't move the other way as easily so this tells you which strand and that tells you which direction. If this was on the other side, then it would go the other direction. So it does depend on that, okay? It indicates which one's the template strand. Again, reading through DNA, we can see these regions and see also where the RNA polymerase is attaching to identify that there is a gene there that's being read by mRNA. And again, there's different promoters for mRNA, RNA, rRNA, and tRNA. One of the keys here is understanding that those different segments that are there is going to tell you. The TATA is mainly for mRNA in eukaryotes and prokaryotes. Okay. So, eukaryotes, we have a little bit more things happening here, okay? Looks a little bit crazier than that very simple drawing that I showed you before, okay? One of the key things I wanted to focus on here And I'm going to share a video in your, when I post this online, kind of understanding where some of these could go and these activators and how they work, okay? All right. So you have the TATA binding protein, okay? That's the one, that's the initiator protein. That's the one that says, okay, here, start here. Attached to it are a bunch of other stuff, okay? What happens is that these are attaching some number of bases away, okay? Enhancing, okay? There's activators. These proteins bind to genes as sites known as enhancers. Activators help determine which genes will be switched on. They speed the rate of transcription as well. And the repressors can do the opposite. Again, these are further upstream from the TATA box, okay? These activators will be here. And the activators here are influencing these co-activators, adapter molecules, that are attached to the TATA binding protein and telling it, is there activators here? Are there enhancers? Is there a silencer to turn this off? So there's a lot of things to regulate our gene expression. What genes need to be turned on here? What genes need to be turned off? A lot of this has to do with chemical signaling in the body that we're not going to be talking about as heavily. But these all are interacting, and if we could influence, say, which ones are activating, which ones are silencing through chemical manipulation of our body, which is what we do with a lot of medications, actually, encourage certain things to turn on and off to encourage the transcription of a certain thing or not. Some medications can do this part, influence actually what's being created, induce the production of a certain protein, repress the production of a certain protein, okay? And that interacts within your own body chemistry to create a medication effect that is beneficial, okay? Again, so these are further upstream than the TATA box, okay? And again, any of these enhancers, these all are working on this gene, but in here there could be another gene and different parts could then affect that, okay? All right. All of that to say, attach here and go this speed, okay? All of this is telling it to do that. So RNA polymerase has now attached, here, and then a little bit further past the TATA box where it says to go, it finds the coding region, okay? So that's a certain distance away from the TATA box. All right. All of this together creates the whole process to figure out where is the gene, how necessary is it, how much are we transcribing, and go. Okay, then we have the initiation. Similar concept here again. So here's a smaller representation of all of those active things going on, okay? Then it, after it has opened this up, okay, it adds again to the 3' end. So it's adding the 3 to 5. So this is the 5' end and this is the 3' end. I already talked about that. We're talking about mRNA here, but same thing will happen in tRNA. This same thing will happen in rRNA. It's just a different promoter and a different RNA polymerase in our bodies that recognize that. Okay. So in prokaryotes, the termination varies. There's what's called row-independent termination, okay? What happens is that there's an adenine repeat, okay? So it goes, ah, and then the RNA reads, you, creating this loop, that chops it, okay? So that's the end of it. The row dependent, that actually induces termination based on the row interacting with a RNA sequence. Okay, so this protein cutting it versus and this section, okay? All right. And then row independent, what happens is that you have these inverted repeats. A, G, C, C, G, C, C, T, C, G, 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 C. And those are here, okay? And then it has this section, okay? Once it reads that, these interact and squish onto each other, making this loop. And then the loop will break off at a certain number of A's. Okay. It slows the transcription, forms a hairpin when transcribed. Then there's this poly A, polyadenylation, signifies the end of a gene. And that creates the okay. So we'll get to the different parts here of independent versus non-independent. I'm realizing I should slow down and stop here. But we'll talk about eukaryotic ones on Thursday, and I will upload what I've covered so far, the termination part, and then we'll talk a little bit more about after the mRNA is made, how is it edited to go out, and then and again, that editing and the interactions in the genes there, what's edited and what's not, and how does the RNA know what to read is really the goal here. What's an intron, what's an exon, how do we figure out what it is, and how does that interact also with potentially creating different proteins based on what's cut out within introns. So we're going to talk a little bit about that because that's important for gene expression. Okay. A lot of information into you guys. Let's take a 15-minute break. We'll come back at 3:35. I'll turn on the Zoom. Have I shared the Zoom meeting link with you? I guess I haven't. I created a new Zoom meeting link. I'll put it here and I'll put it up on there so that everyone can get in. But let me pull this up. This is the repeat one, okay? So this repeat one, we're going to use this one always for our recitation, okay? So this is the key. Okay, so that's our current one. The meeting ID is there. The password is there. I think if you click on it and you go through your own computer and you have an account with Zoom, then you don't have to worry about passwords. Again, if you have issues accessing any of this, we can, I do record them and I'm going to be posting them. So you'll have access to it. Do you guys have any questions about what I just went through? If not, we can stop and we'll go through the insulin-based case. Talk about that a little bit. I encourage you guys in Zoom, please do talk to me. Having me talking at you to a screen that just shows your names is not very interactive to me. We'll try to actually get some interaction this time. Okay, I will see you guys in a few minutes. Take a break. And again, I'm going to be posting this ASAP. Some of this is, I just put it together today, so I haven't had time to edit it and get it in the portion that you guys can see. Okay. Any questions? All right. I'm going to stop the stream now, and again, please go there. I'm going to post this up as well for anyone who didn't participate in the